A very good afternoon to one and all present here today. On behalf of School of Life Sciences, University of Hyderabad, I welcome all of you to the distinguished lecture by Professor Samir Bhattacharya. I would like to invite the respected dignitaries, our Dean, School of Life Science, Professor N. Sivakumar, our Vice Chancellor, Professor B.J. Rao, and today's distinguished speaker, Professor Samir Bhattacharya, to come on the dais. Please come, sir. Thank you, sir. Now I request Professor Srinivaslu Kurkuti, Head Department of Animal Biology, to welcome the gathering.
Hello, very good afternoon. Honorable Vice Chancellor Professor Vijay Rao. Very good afternoon to everyone. Honorable Vice Chancellor Professor Vijay Rao, and then Dean School of Life Sciences Professor Shivakumar, and then today's distinguished speaker Professor Samir Bhattacharya, and all to the gathering. Very warm welcome to this distinguished lecture. On behalf of the Department of Animal Biology, we extend a warm welcome to all of you. It is our privilege that today we have a distinguished speaker, Professor Samir Bhattacharya, one of the eminent scientists in India who has contributed significantly to the progress in the field of type 2 diabetes. And we hear a lot about his research activities in India, which has really created a paradigm shift in the context of pandemic diseases. So with this little brief introduction, I once again welcome you all to this distinguished lecture. Thank you.
Thank you, sir, for your cordial welcome. Now I request Professor N. Sivakumar, Dean School of Life Science, to brief about the school and welcome the gathering.
Honorable Vice Chancellor, distinguished guest Professor Samir Bhattacharya garu, faculty colleagues, student friends, Registrar Dr. Devesh Nigam garu. It's a pleasure to welcome all of you this afternoon and talk about the School of Life Sciences, which is one of the largest school among the 12 schools in the University of Hyderabad. And this school is having 5 departments and several PhD students working in these departments and different master programs that are offered by each of the departments. We have 200 plus master students, and we already initiated the NEP 2020 program, and we are going for the common entrance exam even for the PhD admissions. The School of Life Sciences is one of the vibrant schools in the University of Hyderabad with lot of scientific potential. Every year there is increase in the number of publications, more number of students getting different types of awards, the more recent ones being the Prime Minister Research Fellowship Awards. Many of the life sciences students have achieved this, which is always speaks about the quality of work that is being done at the School of Life Sciences. And we're also fortunate to have a biologist again heading the University of Hyderabad, and he's also part of the School of Life Sciences, Professor B.J. Rao garu. And under the leadership, I am looking forward that we move forward and more strength year after year. Professor Samir Bhattacharya garu is not new to the School of Life Sciences. He has been a visitor to our university quite a number of times, and we are very happy, sir, that each time you come, you spend a lot of time with students and also deliver a couple of lectures, popular lectures and specific scientific lectures. And some of the faculty may be same, but definitely the students vary each time. And I'm sure every batch of students will be benefited from your experience and expertise in the area of research, what you present to them. We are happy again to invite you today for the distinguished lecture series, and we look forward to hearing on the excess of lipid accumulation associated with type 2 diabetes and cardiovascular problems from you today, sir. Welcome again. Thank you.
Thank you, sir, for your kind words. Now I request Professor B. Senthilkumaran to introduce our speaker.
Good afternoon, one and all. Respected Vice Chancellor Professor Vijay Rao, our Dean, School of Life Sciences, Professor N. Sivakumar, and our distinguished speaker, Professor Samir Bhattacharya. It's really indeed honor for me to introduce Professor Samir Bhattacharya before you. Professor Samir Bhattacharya did his PhD from Presidency College, Kolkata. Then he did his PhD from IACB under Calcutta University, and he has received several awards and recognition for his work. He started his work in fish endocrinology and went up to the molecular level in endocrinology, and after that he had shifted his work towards diabetes mellitus, and he had excellent publications on Nature Medicine, which has got excellent citations. In addition to that, Professor Samir Bhattacharya has got trained in University of Washington, Seattle as a Fulbright Research Fellow, and also he also received number of CSIR Technology Award, FICCA Award, and also had the opportunity to do the directorship for IACB Kolkata, where he did his PhD, which is a rare distinction, I always feel. And then he is also a fellow of all Indian science academies. That's another privilege he has got. And in addition, he has been invited for several universities to give invited lectures, starting from Cambridge, University of Tennessee, Tokyo, and also in Waseda. And also, I'm very happy that he is giving distinguished lecture here, so which is again an feather in his cap. And I'm really thankful to him for coming all over the year. And I also thank our Vice Chancellor to immediately agree for this distinguished lectureship upon recommendation from our Dean of School of Sciences. So really I'm privileged, sir, to have you here as a distinguished scientist. And also you are an INSA distinguished scientist, and he has served in INSA on several capacities. Thank you very much, sir.
Thank you so much, sir. Now I request our Vice Chancellor, Professor B.J. Rao, to felicitate our distinguished speaker, Professor Samir Bhattacharya. Thank you, sir. Now I request our Vice Chancellor, Professor B.J. Rao, to address the gathering.
My association with Professor Samir Bhattacharya goes decades ago. I formally welcome you here. It's our pleasure to invite you as a distinguished speaker and to honor you. I met you, I think, few decades ago. After that, our interactions has reduced. But when I met you, I realized that I met probably the best physiologist and endocrinologist of the country. Physiologist cadre is very, very sharply declining. We hardly see physiologists in the community now. It's always a pleasure to hear you and to hear you describe about the physiology of, let's say, macrophages and the insulin resistance, the lipid physiology, and how the endocrine system crosstalks with it. These are all musical words for some of us. So you really explained this very, very well. You taught some of these basics long ago. Therefore, some of your take-homes really stayed with us because your work was so impact-making. So it's again a deep pleasure for us to welcome you here. I am sure all my university colleagues here in the life sciences have been benefited by your presence. I believe you came day before yesterday, so I'm sure you have had good interactions with our colleagues here. So I am now all ears to listen to your talk on type 2 diabetes, to see what new things you have done and how you are going to present the case. Thank you very much.
Thank you, sir. I now invite Professor Samir Bhattacharya for the distinguished lecture. Sir, please.
Our Honorable Vice Chancellor, Professor B.J. Rao. In fact, Professor B.J. Rao known to me for a long time. And as he said that he heard my lecture at the, where he was earlier in the, so I have heard his word, he has probably forgotten. His lecture I have also heard. He gave wonderful lectures in that particular institute. And Professor Shivakumar also gave a good introduction, and all of you came here. I'm so happy to see the people here. But I'm especially happy to see Professor B.J. Rao here, who is the Vice Chancellor. One day we'll be able to find what a wonderful person he has. That kind of, academic person, plus at the same time a good administrator is very rare. Professor Rao is here. I'm so happy to see him here today. So anyway, all of you with the best wishes for especially to Professor Shivakumar and Professor A.S. Raghavan and Professor Saltin Kumaran. So I am really grateful to your Vice Chancellor, to all of you, because you have given this opportunity to tell you whatever we are doing in relation to the very critical disease. A critical disease in the sense this disease is pandemic, going all over the world. But till now, this disease has not a medicine which you can tell it will make it cure. No, there is no medicine like that. And all the medicines which are coming up usually most of them have adverse side effects. And that adverse side effects in some cases it is coming to the kidney, so it is very bad. So I'm telling you some of the drugs which are coming to the market for this particular disease, which is, we are all becomes a diabetic. Diabetes is the disease. Medicines are so much in the market for diabetes. I'm telling you for a few instances, sitagliptin, vitagliptin, these are in the market, and these are going on profusely into the market. And now that they have found this sitagliptin and vitagliptin, which is actually responsible for one of the things, that is insulin sensitivity increases. That is true in one sense. Another sense, it is bad because it is going to the kidney, and in the kidney it increases the pancreatin. So as a result, creatinine is increased in the kidney because of these two important medicine, what is sitagliptin and vitagliptin, which is important for insulin sensitivity, but at the same time, it has adverse effect also. This is coming with the other drugs into count. Sulfonylurea is another drug which is coming into the market, which is in the market, but very slow in going the disease to tail. In fact, we never find a very satisfactory medicine for type 2 diabetes. And type 2 diabetes is something which is also related to the problems of cardiovascular system. And why it is a problem in cardiovascular system, that I will describe. But important thing is that this disease all over the world is going on in the same way, but till now, not a single medicine, either from the Europe or from America, is giving out the rescue. Yeah, sulfonylurea is one of them, which is not very much, no negative effect or side effect, but it is a very slow medicine. It does not work very much. So as I already told you, sitagliptin and vitagliptins are actually winning the market, but they have a problem. One thing is, in the kidney. And kidney is a very, very sensitive area, as you know, because once there is anything wrong with the kidney, there is no drug until unless you just have a surgical operation or something, which is not good. So with this, I want to tell you that we are hectically, every part of our world is looking forward. If something comes out, diabetes type 2. Diabetes type 2 is increasing like anything, and it is there without any proper medicine in the market. So I request you, all of you, this university is one of the best university in our country. I'm very happy that Professor A.G. Rao is here. Now, one thing is, and also that Professor Shivakumar attended. Professor Raghavendra is my good friend for a long time, and Senthil Kumaran is also there. But I came here earlier also. I'm sure Hyderabad University is doing something very good. And they will also give their concentration towards these two diseases. One is two health science problems. One is this diabetes type 2, this is number one. And number two is cardiovascular problems, which are still remain, a little bit a problem too. And it is so much in our population. So coming to this, I want to, may I have the next slide? I want to tell you that okay. So you can see that this is a schematic diagram. What you find here, where does the glucose goes? It is not very difficult. Glucose come into the blood, and if glucose can enter into the cell, especially the skeletal muscle cells are the highest number of cells in our body, because it is about 72 to 75% of tissues are skeletal muscle. So if that is stored it can enter into the skeletal muscle cell, besides the liver and other places, there is no diabetes. So only thing is it has to enter into the skeletal muscle cell. Why it is not entering? This question is the most important question that people usually ask, and why it is not entering. The reason is, for its entry, I am coming to it. Please remember one thing, that this is the major problem, that glucose from the blood is not entering into the skeletal muscle cell, and why. And that why should be answered. And that why is still today is not answered properly. So that's why diagnosis and its medicine is not very suitable till today. What you see here, that after food, usually you can see the glucose is going up to this till 120 minutes, and it fall down at 120 minutes. You can see it is come back again. This is because the insulin also increasing from 35 picomoles to 125 picomoles to this direction, and insulin retards the entry of glucose into the skeletal muscle cells, so it comes down. So this is the usually postprandial, means after taking the food. So after taking the food, it goes up. Insulin also goes up from 190 to 300 picomoles, and ultimately it goes down to the baseline at 120 minutes, that is 2 hours after. This is called that, this is the picture. But what happened, next slide, what happened in the case of diabetes is this: if the glucose goes down to the cells like adipose tissue and skeletal muscle, then everything is okay. But if not, next, if they do not go from the blood circular system to adipose tissue and skeletal muscle, as I told, 70 to 75% in skeletal muscle, and about 25% is adipose tissue or adipocyte in the red. And then if it cannot, then obviously it will remain in high level. It will remain in like that. From 120 is the maximum normal. Then it goes up, as you know, 200, 250 like this. So as a result, it will be a diabetic subject. It may be type 2 diabetes, which is 95 to 97%. Type 1 is very less. But this is a disease which is continuously increasing. But there is no proper drug, no proper medicine, there is no proper treatment. And till now it is what it is. Another thing, bad thing is that it is also creating a problem with the heart problem, circulatory problem, and other problems, including the problem in liver and kidney. So this is the high sugar level is dangerous for us, but we are not able to maintain, can't do anything. So we started with this, our work, and I want to tell you what we have found is something different than what Europe and America has found. So it's something different. So I'm telling you, there we will soon publish it. There is no doubt to publish some portion of it, but not all the things. Next slide. What is important? This is the insulin receptor, which is a polypeptide. As you can see, the 2 alpha cyanide to beta cyanide. Insulin binds here, and after insulin, when insulin binds here, these are the tyrosine which has again phosphorylated. It is not properly phosphorylated earlier, but once insulin binds. Next slide, you did. So insulin binds here. So if you see that tyrosine is phosphorylated, there is tyrosine here, and so all this tyrosine is being phosphorylated. And once tyrosine is phosphorylated, next slide, next, there are 2 carrier. One is insulin receptor substrate 1, IRS1, IRS2. They pick up this phosphate from there and carry it to the different kinases. So you know the kinases are the enzymes which actually carry the phosphate. So phosphate carrier. So kinase carries this phosphate which came here, and this phosphate comes here because it is an energy. Of course, this energy comes here, and energy goes with this, ultimately going into this direction. Next. So these are kinases, all these kinases, this is PI3 kinase, this PTK1 kinase, then ACT or PKB kinase, all these kinases are carrying this phosphate from here to here. Why they are carrying this phosphate? What is the necessity of carrying this phosphate? The reason for carrying this phosphate is that to make glucose entry. Why? I'm telling you that. Next. Phosphate is coming with ACT or PKB, and you can see these are the glucose transporter. And glucose transporter is remaining in the cytosol of the cell. So glucose transporter is GLUT2, GLUT4. There is GLUT1, 2, 3, 4, but GLUT1 and 3 are not very active. GLUT2 and GLUT4 are the glucose transporter. And these GLUT2 and GLUT4 are here. That means there is a bundle here. But when they are here, they need to go to the membrane to carry the carry the glucose. So GLUT is the glucose transporter, and GLUT2 and GLUT4 are the major glucose transporter, and they will go to the membrane. They cannot go without an energy. So if they cannot go without an energy, this phosphate should be given there. Actually, this kinase is doing the same thing. So phosphate is coming from here after the binding of insulin, and this message is going on with the kinases and comes here, and ultimately when it comes here, it gives this phosphate to the GLUT2 and GLUT4 who are here. GLUT2 and GLUT4 goes to the membrane, allow the glucose to come. If the glucose comes and this system goes like this after insulin binding, there is no diabetes. Diabetes is the condition when the glucose remains in the blood. Glucose does not go into the cell. This is simple, appears to be very simple, that glucose from the blood will come to the cell. This is the way the glucose comes to the cell. Now you see, the whole world is threatening by this disease, because this glucose is not only spoiling your liver, not only spoiling your heart, this is also spoiling your kidney. So as a result, this glucose is a dangerous disease now today, but even if it is such, we are not able to control it. Now, next slide. Next slide. What happened in the case of the energy actually is a metabolic disease. What happened in the case of the energy, you see the energy is being intaken by your food, by your nutrition, you get the energy. Energy is also stored, but energy is also expenditure. If these all remain in good track, we called it that energy homeostasis. That means everywhere it is being maintained. This taking of energy, storing of energy, expenditure of energy, if they go all right, then fine. They call that energy homeostasis, but it may be disrupted. And when it is disrupted, the condition is for why it has been disrupted. I told you that energy comes into the skeletal muscle tissue, also come to the adipose tissue or adipocyte. If adipocyte can take them all right, then everything is all right. But something happened to the adipocyte. And this is what? Excess of lipid make adipocyte store like this. So you can see the storage of lipid or fat into the adipocyte may be excess. If it is excess, the cells are not normal, they are abnormal. So what happened? That energy homeostasis is maintained because cells are normal. Cells are not in this way, they are changing their shape because of excess of lipid being deposited, so energy homeostasis is disrupted. If it is disrupted, then all these metabolic disease sets in. Exactly happens in the case of the diabetes and other cases also, that adipose site is the most important tissues here. These we neglected for a long time. Everything we take, but we are not thinking about the fat cell, which is adipocyte. He stored the fat. The storage is very important. I am coming to it, not only storage, they induce also something. So next slide. One thing I am telling you, it is very interesting to tell you today, that this insulin resistance or insulin inactivity or insulin problem coming up because of the reason that mitochondria goes down. Why mitochondria goes down? Mitochondria number, population of mitochondria goes down. As you can see from here, that mitochondria goes down to here from the normal mitochondria level. So this is a normal level, which is called, the normal level remains with the standard diet. With the standard diet, it goes like this. But you take high fat diet, this level goes down, mitochondrial level goes down. Mitochondria is necessary. Why? During the diabetes, mitochondrial level goes down. So population of mitochondria goes down. And why population of mitochondria goes down? That I am coming. But if mitochondria goes down, then what will happen? Then energy will not go to GLUT2 and GLUT4. So GLUT2 and GLUT4 will suffer from energy, and they cannot go to the membrane. If they cannot go to the membrane, glucose will not come inside. Matters appears to be very simple, that if there is energy, then there is no problem, GLUT2 and GLUT4 will go to the membrane. But if it is not, energy comes from mitochondria, as you all know. If mitochondria go down, energy will be little, ATP will be less. If ATP is less, then you cannot use that ATP for conversion there. So that happened. In the case of the high fat diet, this is exactly what happened, that mitochondrial level goes down, as you can hear, HFD is here. And therefore, when it goes down from normal like this, SD is the standard diet, and then mitochondrial level goes down. We found, we call it LAS. This is a plant, Leucas aspera. This plant is available in the northeast. This plant is available in the case of the in the area of the Arunachal and in the area of Assam. These two are there. In the border, there are some kind of cultivations, and these are nothing but small herbs. And this Leucas aspera, tribal people, they take it. They take it for their some kind of fever, some kind of thing which is not good, they take this. When we came to know that they are taking it because they have some fever, something bad in their health, they take Leucas aspera. It's like a herb. It is a herb, small herb like this, and they take the leaf. When they take this, they are feeling bad. So we collected this plant, Leucas aspera, extracted it, and see what is happening. We are using this Leucas aspera, but not Leucas aspera. Later on, I will show. Now, four people. One is the Shubhan Dajgupta, another is our the Jaishankar, Parashuram Jaishankar, who is Dr. Parashuram Jaishankar in IICB, and we have another person which is working there, which is name is Sungar. So we all are working together, and Jaishankar found it is actually not only the Leucas aspera, but the compound which is actually working. I will come to it. So if you treat with the Leucas aspera, you can see that this mitochondrial number again increases to this level. You can see it is inducing the mitochondrial level up. So once it is going up, then everything is normal. You can see the QPCR, the mitochondrial level goes down, again is going down with the Leucas aspera. You can see here. But not Leucas aspera, it is the compound I am coming to it. So you can see mitochondrial level come up. Once mitochondrial level comes up, then blood sugar level also goes normal. So this is happening here. You can see that fatty acid oxidation and etc. I will not go further. But one thing I am telling you, mitochondrial mitochondrial function goes up. So we get the ATP level all right, as you can see, that if you use in increasing concentration of Leucas aspera, not Leucas aspera, the compound, I will come to it later on, after which has been purified and structure has been made. So it goes up, and you can see the mitochondrial level comes to normal. If mitochondrial level comes to normal, you'll be surprised to know if the mitochondrial level is normal, then again glucose level, high glucose level in the blood goes down, so your diabetic condition is being taken care of. Next slide. Here we want to tell you one thing, that is one thing. So about the GLUT4, which is I already told you it is a glucose transporter. May I have a glass of water or something? Oh, it is here. So I'm sorry, I'm a little bit. So you can see that here we have wanted to test one thing. Among the lipid, among the fatty acid, the most obnoxious fatty acid is palmitic acid. I was discussing this matter with Shivakumar. Palmitic acid is the most dangerous fatty acid. It is a C16 only, carbon 16. But arachidonic acid has carbon 18. The number of carbon may be more, but palmitic acid is most dangerous fatty acid among the all fatty acid. We checked all different kinds of fatty acid to see which is the most dangerous fatty acid, in the sense that they can cause the coming of glucose into the cell being prevented by palmitic acid. Palmitic acid is why palmitic acid doing it, I am coming to it later on. But one important thing is you can see when palmitic acid is given, then GLUT4, even if GLUT4 is being given the trigger, but they are not there. GLUT4 usually come after the glucose comes, go to the membrane, as you know. It is not going to the membrane because of the palmitic acid. On the other hand, if we treat with the LAS and Leucas aspera, that means you can see that GLUT4 goes to the membrane. This shows that palmitic acid, fatty acids are obstructing GLUT4 to go to the membrane for carrying the glucose. And fatty acid do it actually, it is not exactly fatty acid, I will come to it again. But this is the thing that I hope you have already got the message. About the sensitivity, it's not necessary to tell all these things, how the sensitivity comes in. You can see that if you give high fat diet, the sensitivity of the glucose tolerance test, you can see that it goes up. But if it is not clear, glucose is not clear from the circulation, but glucose is clear from the circulation, you use the Leucas aspera. So Leucas aspera is giving you the against doses, against the lipid, against the fat, Leucas aspera is working. That is quite clear from here. Next. Here you can see one thing, very interesting. So these two, one is the diabetic mice, diabetic plus obese mice. This is DBDB mice, this is genetically modified. And you can see that here, one thing is coming here as fatty. What is this fatty? Fatty A. It was earlier thought by many people that it is the fatty acid which is obstructing the insulin activity. Fatty acid is, if deposited much, then they are binding with some other membrane receptor. They are called TLR4 receptor, and then fatty acid is obstructing the insulin function. That means the fatty acid, ultimately you can see that a new thing has come up which is called as fatty A. Actually, we found one thing. That means it is not the fatty acid which binds. Fatty acid is not binding with the TLR4, which is not true. People thought fatty acid. We found one thing, that is not fatty acid which is binding, but fatty acid in the adipocyte is inducing a protein, glycoprotein, and that protein is fatty A. So fatty A is being produced at the adipocyte, adipose tissue here by the fatty acid induction, and this fatty A is responsible for one thing, that they are responsible for obesity and also for the diabetes. So it is a diabetic mice with obesity, and you can see the huge fatty A is being formed. So it is not the fat or fatty acid which is doing the harm. It is the fatty A which is doing the harm. So this knowledge came later on by our laboratory later on, and many people objected later on, everybody accepted. It is true, it is not the fatty acid, Fatty acid induced in adipocyte by a factor, a protein factor, glycoprotein factor, which is fatty A. Now, fatty A is responsible for this. So, fatty A is not allowing the glucose to go into the cell, not the fatty acid. If I am clear, you can see that in DBDB, this is the thing that you remain in the glucose level goes high. So, this is what I want to tell you, that here also it is not DBDB mice, but it is high fat diet induced obese diabetic mice. Same thing occur here. You see the fatty is more, it becomes diabetic and obese. Same thing here, you can see the serum fatty A is much higher. Serum fatty A is much higher and it is diabetic. So, now it appears fatty acid is not the culprit. Culprit is fatty A. So, now whole idea is changing. So, it is taking fat is bad, that is true. But fat is not directly doing the diabetes, this fatty A is doing the diabetes. So, till now, unfortunately, there is no medicine for fatty A. But one thing is, fat, if it deposited is high number, they will produce fatty A. This is the thing which I will like to tell you, that same thing is here, same thing is with DBDB mice and fatty A. Next slide. So, here you can see that one thing is excess lipid produce what? Excess lipid express the fatty A gene, and fatty A gene ultimately produce fatty A protein, and this increased fatty A protein is being circulated, and fatty A protein binds to fatty or FFA, fatty acid, and with this binding they come next. With this they binding, but fatty A binds to the TLR4 receptor, not the fatty acid. You can see here that fatty acid does not bind, it is fatty A which binds. But fatty acid is necessary because fatty acid induces the gene of fatty A in the adipocyte. So, fatty A is actually responsible here for the functioning of the cell. Here it is adipocyte, hypertrophied adipocyte means yellow portion that you see, lipid is accumulated here. But this lipid does not do any harm. Actually, the harm is affected by fatty A. Why? When fatty A binds here, next slide. When fatty A binds here, they activated the transcription factor NF-kappa beta, and this produces, next slide, pro-inflammatory cytokines. And these are the pro-inflammatory cytokines, which is IL-6, IL-6, IL-1 beta, and TNF alpha. These are the pro-inflammatory cytokines. This comes out by binding of the fatty A to the TLR4 receptor. So, this is the thing, fatty acid increase, and it increases in the adipocyte, it induces the gene of fatty A, and fatty A in turn is coming out from there, and ultimately binds again to the another adipocyte. So, as a result, it induces the activation of the transcription factor NF-kappa beta. So, this becomes responsible for producing this pro-inflammatory cytokines which comes out. What does this pro-inflammatory cytokines do? They actually makes the insulin resistance. These pro-inflammatory cytokines are responsible for insulin resistance. This is where we are stuck off. Why? Because there is no drug which can take care of pro-inflammatory cytokines. So, this is, next slide. So, you can see that pro-inflammatory cytokines are produced in downregulation of PGC1 alpha, which is, I am not going to this direction. This is actually for the lipid mobilization, they act very much. They are downregulated because of them, and also inhibition of the mitochondrial biogenesis. So, pro-inflammatory, I told you that mitochondrial number goes down. That means mitochondrial biogenesis, this is the term, and bioenergetics, these are the two portions of the mitochondria. Biogenesis means increase of mitochondrial production. Bioenergetics means that when mitochondria produces ATP, right? If these two things are working perfectly, there is no diabetes. There is no problem of the diabetes. But happen what? These are not the fact. If what happen? If this pro-inflammatory cytokines comes out as a result of the fatty A, not fatty acid, but fatty acid produces fatty A. Fatty A induces the genes of this pro-inflammatory cytokine, and they are coming up. And when they came up pro-inflammatory cytokines, they are responsible for the again inhibition of mitochondrial biogenesis and also bioenergetics. That means mitochondrial number reduces, mitochondrial ATP production reduces. These two are produced by fatty A. Now, what comes before us? Till today we know it is fat. Till today we know it is fatty acid. But now it is quite clear that it is the fatty A, which is a glycoprotein, which is responsible for this kind of factor, which is pro-inflammatory cytokines, and they are responsible for insulin insensitivity. Why? Mitochondrial level goes down, which is actually necessary for it, and mitochondrial biogenesis is being affected, and mitochondrial bioenergetics for producing ATP yield also decrease. So as a result, what you find? That your energy is going down. In all cases of the diabetic patient, you will find one thing, that an energy is lost. Energy is not coming up. And the reason is this, that they could not produce much more ATP. Mitochondrial biogenesis goes down, mitochondrial bioenergetics for producing ATP also goes down. Next slide. If I am clear up to this, I want to tell you one thing. These are the liver cells, okay? So what happened here, and from the liver cells you see that what is coming up here is again the fatty A, which is taking the fatty acid with it. You can see these are the fatty acid which are binding with the fatty A. Fatty A are given yellow, and the fatty acid has come from liver. It is been secreted. Why? If liver accumulates fat more, fatty acid more, fatty acid ultimately goes out from the liver, and around the liver there are fatty A which are roaming in the blood, and ultimately they take up the fatty acids. These are the fatty A. They bind into the TLR4 receptor, and TLR4 receptor goes out pro-inflammatory cytokines, and pro-inflammatory cytokine produces insulin resistance. This is the story. The story is liver also add to the diabetic condition if liver is being with the excess of fatty acid. Now this fatty acid comes with the binding with the fatty A. Same thing comes here. Pro-inflammatory cytokines are too many. All these pro-inflammatory cytokines ultimately reduces what? Reduces IRS. IRS, insulin receptor substrate, is taking the phosphate, if you remember, from the insulin receptor. But IRS is being inactive because of these pro-inflammatory cytokines. So pro-inflammatory cytokines goes and binds with the IRS1 and IRS2. Once they bind there, they cannot take the glucose, they cannot take the phosphorylated from the phosphate. So if they cannot take the phosphate is not goes to the via kinase to up to the GLUT4 and GLUT2. So this is how the GLUT4 and GLUT2 cannot go to the membrane. So glucose is not coming out. This is the story. If this is the story, on the background of this story, you don't find any drug in the market. So as a result, we must get drug which is responsible for decrease of fatty A. People have understood all over the globe. All the fatty A have produced from India. I mean this knowledge, but they now understand that without reducing fatty A, we can't manage diabetes. So this is the story. Next slide. I'm coming to the end of it. I know that much of it is here. You see, these are the fatty acid, and this is what is fatty. So fatty acid binds to blood in the blood vessel. Once they bind with fatty A, they come out of the blood and come to the cell. This is what I want to tell you. You already told it, but you can see that this is the blood vessels where this is the fatty acids which are red blood, and this is fatty A is in blue. They bind together. Now we have found this, that ultimately they are bound. They come out of the vessels. They come out here, doing the same thing pro-inflammatory cytokines. So what you see, that flowing of excess of lipid within the blood also is not good. It is binding with the fatty A in the blood, and they come out. So question is that people are asking sometime, how you are telling that it is coming into the blood? Because fatty A molecular weight is this, about 32. Is it coming from the blood to the out? We said yes, because they have the membrane receptor which is taking them. So now next slide. So this is the same thing. I will not go further detail. We have already discussed how they are bind. If this is binding, we have analyzed. This is fatty A. In this binding, these are the different kinds of glucose, aspartic acid. All these are here. They ultimately bound to fatty A, and they bound here, and ultimately the message goes into the cell. So this is in detail. We've already worked out that how fatty A is bound into the TLR4 receptor. So TLR4 receptor binds with this kind of machinery, which I am not describing here. You can't see all of them. So, but only thing is, this is how the receptor works to take the phenytoin. Next slide. Coming to the end point of it, most important thing is, if that happens in this way, then phenytoin A, is it responsible for the inflammation? Because inflammation is the disease. Actually, diabetic is an inflammatory disease. And how it is occurring like this? Who is actually doing this inflammation? How this inflammation occurs? You see, this is the macrophage, and with the macrophage which is anti-inflammatory is known as M2, macrophage which is pro-inflammatory known as M1. So M1 is pro-inflammatory, M2 is anti-inflammatory. These two macrophage, I am telling why. Reason is why. Are they connected with diabetes? Yes, they are connected. How they are connected? Macrophage is connected with diabetes, people will laugh. You are telling that macrophage is connected with diabetes? We have shown it. Now the paper will go ultimately. What is important, I am telling you this, that you see the adipocytes are here, FA is here, phenytoin A is here. Now from there you can see one thing, that these adipocytes, they are produced fatty acid. This fatty acid comes out, and phenytoin A being produced, phenytoin A is given, phenytoin A is resulting M2 conversion to M1. As I tell you, M1 is pro-inflammatory, it is anti-inflammatory. Only thing what I want to tell you, that if M1 concentration increases in adipose tissue, they enter into the adipose tissue, adipocyte. If M1 concentration increase, then means that fat are entering into the M1, not into the M2. So fat is loaded in the adipocyte. They are coming to M1, not to M2. So M1 becomes more important than not M2. M2 is not doing any inflammation. This is doing the inflammation. So I'm not going to all this. You see, MCP is chemoattractive protein. TNF alpha, you all know, it is increasing. It's because of fatty. MCP is also increasing. What is MCP? MCP is monocyte chemoattractive protein. This protein increases. Why? This protein carries out the phenytoin A inside. Increases because of the phenytoin A. Phenytoin A is the main culprit. Next slide. What I'm telling you, that once this M1 macrophage is taking more lipid inside, they are being foam cell. So macrophage is converted into a cell which are known as foam cell. And foam cells are cells which are going through the vessel. They are being attached to the inner wall of the blood vessels. It may be artery, it may be vein. If they are stuck to the artery there, they produce atherosclerosis. What is atherosclerosis? Is the stiffness of the vessel. If the vessel is stiff, what will happen? There will be some crush with the blood, and there is Hegeman factor or factor 12 which produces clot. That means fibrinogen becomes fibrin, they form a clot. And that clot is affected because in the case of the diabetes, this M1 increases. M1 increases because M1 is being induced by phenytoin A. So as a result, phenytoin A is doing the same thing. You can see here, these are the oxidized low LDL, meaning oxidized low density lipoprotein enters into the foam cells. So foam cells are more into the blood vessels. They are on the wall and they produce atherosclerosis. If atherosclerosis is there, you can easily understand there will be friction of the blood. So that's what I am telling, that how diabetes is related to cardiovascular problem. So this is vascular problem where atherosclerosis has been formed. This atherosclerosis is formed coming from lipid to phenytoin A to M1, M1 to foam cell. Fatty acid to, as I told you, fatty acid, phenytoin A is being increasing the M1 and giving it the passage for the more loading of the fatty acid. Now, this within the fatty oxidase is the most important, and if it is increases, these foam cells becomes very sticky into the wall. You remove the oxidized LDL, they are not sticking to the wall. So we found one thing, that oxidized LDL means low density lipoprotein. So if oxidized LDL is removed, if you can remove, then there is no stiffness. Stiffness is produced by oxidized LDL. So what a wonderful finding it has Shumon and other people have worked with me and they did it. This wonderful work. This is the thing that foam cell form. One foam cell forms, so you can see from starting from there, diabetic patient come to these foam cells, and foam cells are on the wall, atherosclerosis occur, as a result blood flow becomes restricted. And if there is a friction, they will produce the clot, and that clot is factor 12, which produces the clotting here in foam cells. You see these foam cells. Now we found a plant as a cave, Leucas aspera. This is the Leucas aspera plant, which is found in Arunachal and Assam border, and this plant produces a compound, we call this anisofolin A. It is the Purusharam Jaishankar has already given this structure, and we found it to be clear. Anisofolin A does what? Anisofolin A binds to foam cell. When it binds to foam cell, oxidized LDL is released. If oxidized LDL is released, they do not remain as foam cells, large cell. They become stringent. They again become smaller. So atherosclerosis becomes removed. So once you removed by this anisofolin A, I'm telling before you, we have to patent it. Otherwise, it is go to the company, then we are lost. So thing is that this is not very easy to come down from here to here. This is anisofolin A, removing the oxidized LDL, foam cell becomes reduced, and when foam cell reduced, they are coming out from the wall, they produce again the passage. You can see that oxidized LDL, anisofolin A, here you can see that foam cells are removed. So when the foam cells are removed, next slide. So this is the cause of high BP and blood clotting. So as I told you, foam cells are there, you can see the passage of blood vessels is restricted. If the passage of blood vessels is restricted, there is every time there is a question of being the binding on the wall and friction. If the friction is there, then factor 12 or clotting of blood occurs due to the fibrinogen conversion to fibrin. So this clotting occurs at that same time. But otherwise also, foam cells binds to the wall, prevents the atherosclerosis. So atherosclerosis is not good, as I already told you, because little blood goes. Allow little blood to come, friction is always occurs. So that's how the cardiovascular problem is related to the diabetes, related to this kind of things. So you can see that after using the anisofolin, they are again reduced. All lipids are being pumped out, oxidized LDL comes out, they are again normal. So anisofolin A does a very good work in this direction. As I already told you, that look at from where we started. We started with the diabetes, and diabetes comes from lipid. This is true, not from exactly from lipid, but from the phytin A. That idea, which was earlier before, that is lipid, it is lipid, because lipid induces the gene of phytin A. But our main culprit is phytin A, not the foam cells. But foam cells are not the fatty acid. Fatty acid is responsible for phytin A. But our later work is made, later hazard is being done by the phytin A, not by the fatty acid. That I told you. So as you can see that foam cells, after you use an anisofolin A, the lipid goes out, oxidized LDL goes out, they become again the foam cells are reduced, so they are nothing but macrophage. So macrophage come back. So these are all macrophage. These macrophage converted into foam cells. They again come back to macrophage. But area of the volume of the blood to go remains clear. So this remains clear. I don't know anyway whether it will work. Yeah, anyway, it will work. You don't have to come. It is follow. So you can see this, that this is how anisofolin A we have, which is already in structure, we are going to patent it. But important thing is, it is giving enormous value to diabetes. Now, this is what we are looking for, that if fat and phytin A could be taken care of, then obviously diabetes will be coming back to normal. Next slide. So this is the end point of it. This is the last slide, right? Is there any? No. So I must tell you how many people worked with me. This is not there. The last slide must be there, but I forgot it. So it is the Shuman Dasgupta who is working from Tezpur University. He is the person who is going to the border area of the Assam and Arunachal, where these things are available. This Leucas aspera is available. He is doing all these things. He is supplying these. Then Parshuram Jaishankar, Dr. Parshuram Jaishankar in the Indian Institute of Chemical Biology, IICB. He is there. He is working with us. And a person who is working in a very long distance, is only in the college. They come to the university now, it's a state university, that Border University. He is doing the rest of the work where the blood vessels can be remain together and giving the this beautiful plate he has done where blood vessels can be there. You can see the blood vessel, how it is working, what it is doing, that particular thing that he has devised, and he is working with that. With this, I must thank all of you for this, and I am again very, very grateful to the Vice Chancellor of this university, Professor Rao, and also Professor Raghavendra, and also Senthil Kumaran, Professor Senthil Kumaran, and also Shiv Kumar has already praised me. So all of you, I convey my heartfelt thanks to all of you. If there is any question, I will be really very happy. If you, I don't know, will you allow question? I will ask my Vice Chancellor whether he will allow the question. Okay. Yeah, please. Yeah.
For quite some time, I also work on insulin signaling and insulin resistance, type 2 diabetes, and anti-diabetes. Excellent talk, sir. You deviate your whole your story towards 15A. I was wondering whether why you have not looked upon PKC pathway, which is really a drug. I mean, the main thing for the insulin resistance is volume of diet, ceramide, etc. Totally, I'm sure you must have seen it, but I was wondering why you didn't get any data on it. Because it totally deviated your data towards 15K. So if you have some data, anything, comment, happy to know. Thank you.
I will give you all the data, no problem about it. About the PKC, we have worked a lot. And PKC beta is one of the beta which is very active in the case of the foam cell. So PKC have worked thoroughly, but I have not entered PKC because it will be longer for me to give you. Yes, PKC have worked, but PKC beta is a dangerous PKC. It is actually functioning the foam cell. So we have worked with PKC beta and also PKC alpha. That is true, but I have not told about PKC because story will be longer. So, but if you want to know, I will definitely send you the data.
Thank you.
So.
Just I have the one doubt. You use the DV mice, and where you see the 15A is high. What about the normal when is the leptin have the proper receptor? What happens in general population? I don't think that people have this leptin receptor deficient and mutant in diabetic. I don't know what is the percentage. But your case is showing in that when DV case, you have the very high fatigue, but normal is not. So does any role in the leptin resistance related to the insulin to correlate this converting to the diabetic case?
Yeah, no, I'm telling you, leptin is not here actually, leptin is in the brain. And leptin goes, as you know, the leptin is giving the signal from here. So it is not necessary that they will give the signal from here. Why? Because in the case of the obese, it is the deposition of the fat. And this fat is deposited where? Is majority fat deposited in the adipocyte. And when the fat deposited, fat is directly not doing any harm. But fatty acid, especially palmitic acid, we have seen 5 fatty acids, which are really doing the things in the fatty gene they are expressing. So they bind with that particular gene and they're producing the gene expression. And this gene expression comes from fatty acid, but they're producing fatty gene is expressed by the fat, by the lipid. But leptin is, it is because of the hunger, the leptin is functioning because of hunger. So leptin is not functioning here because they are not hungry. These people, they are not doing food for this. They are lipid accumulated in case of the lipid mice. Yeah, they take a lot of food, that is true. But we always fed, high fat diet. So once you give the high fat diet, they are deposited there. So function of lipid, function of fatty acid is to induce the fatty digestion. Earlier idea was different. Earlier idea was that it is the fatty acid which is responsible for going into the insulin resistance and not anything else. If fatty acid does it, fatty acid binds to TLR4, fatty acid binds to there, and they are producing the inhibition of the insulin resistance. Now we found no, fatty acid is not binding. It is not fatty acid, it is fatty acid. There is a tremendous difference between the two. It is fatty acid is there, but main function is performed by fatty acid, not fatty acid. Leptin is for the hungry and all these things, etc. We sometimes forcefully give the feeding that is fatty acid. And we also tested different fatty acids. Saturated fatty acid, among the saturated fatty acid, as I told you, it is the palmitic acid which is dangerous. Of course, arachidonic acid is also, but palmitic acid is the more dangerous, not the unsaturated fatty acid.
Yes, sir. Very nice presentation, lot to learn.
Thank you.
And I had two questions, but one you just answered. I was just wondering whether what happens with other fatty acids and how much levels of fatty acid go up with different fatty acids, because we know various fatty acids have various atherogenic index, so the way they can form atherosclerosis. But I think 16-0, like you said, palmitic acid is the most atherogenic of all. I had one more comment in terms of the graphic which you showed, where you said that free fatty acid binds to fatty acid. But under physiological
Free fatty acid bind to fatty acid.
Under physiological condition, most of the times fatty acids are not free. They are either bound to albumin or they are esterified to CoA, depending on what the fate of fatty acid is. So, under what condition do you think fatty acid will bind and will be fatty and fatty acid complex is available in the blood? That's what I just wanted to know.
Your question is all right, it's a good question, but I'm telling you, coming up the fat from the adipocyte is doing by the lipase. So if the lipase activity increases, then obviously the accumulated fat comes out as a fatty acid. And that comes out when there is a lipase activity increases. When lipase activity increases? Lipase activity increases when there is an increase in the lipid accumulation in the adipocyte. And now the question is, if excess of fatty acid comes out, they are on the float. They are not on the wall. They are on the float. We are doing with the excess of lipid, not with the normal lipid. You are absolutely right. Normal lipid always take the wall out somewhere. They are called the ectopic deposition of fatty acid. We call it ectopic deposition. Ectopic deposition may be in the wall, ectopic deposition in the liver, ectopic deposition may happen anywhere. You are absolutely correct. But if lipase activity increases, free fatty acid comes out. Yeah, please.
What is the fate of the oxidized LDL after releasing from the foam cell?
Just a moment. No, somebody from here. Okay.
What is the fate of the oxidized LDL after
Fate of
the oxidized LDL?
Oxidized LDL.
After releasing from the foam cells when you treat it with your drug, because it is ultimately staying in the blood. Any other cells are taking this one?
No, actually oxidized LDL, when you destroy the anisophyllin A, it does not come outside, they are dissociated. So LDL goes out, oxidized level goes down. So it is oxidized molecular oxygen, it is separated when you do the anisophyllin. Anisophyllin detach them from one another.
What about along with oxidized LDL, other fatty acids also form the foam cells, like palmitic acid, acid. What about your drug play a role in that?
It is not my drug, it is all of us drug. Anyway, what I am telling you, it is not drug yet. We have just used it. And we have found it by, as I told you, Jaishankar actually isolated it and structured it. But thing is that what actually does biogenesis LDL is that we know they are separated. We have not come to the level of this, that how this is being again attached or again doing something. This level we have not come. We are coming out.
This it will affect the other fatty acid induced foam cells also. For example, palmitic acid also form the foam cells. Same action is there or
no? Yeah, foam cell actually we have found 5 saturated fatty acid are actually not others. Among these 5 saturated fatty acid, one is the most majority fatty acid as with palmitic acid, arachidonic acid is another. All these are actually, and some of the fatty acids are there. But majority role is playing by the palmitic acid and arachidonic acid. First palmitic acid and then arachidonic.
Hello sir, good evening.
Yes, please.
Sir, I have a small doubt. Sir, in one of your slides, you shown that you extracted leaf and you found that it is identified as an active compound. Sir, plant secretes many secondary metabolites, where several are toxic as well as active. So, sir, how you differentiate between the toxic ingredients as well active? That how you found that whatever you extracted is active compound? Because plant secrete toxic as well compound.
Plant secrete, but this anisophyllin A is coming from the Leucas aspera. The plant is Leucas aspera. This plant is an edible plant, and the tribal people eats this plant. So it is not a toxic plant, first of all. So one thing is the tribal people takes this plant, and we came to know from them, if they take this plant, their health remains all right. This is it is just an information coming from them. This kind of information is very good. Once you get this information, you search it, whether it is true or not. So we went there, we found they take it. Leucas aspera has been taken by them. If you go, it's taken by them. So that's why we thought if they feel happy, if their health is good, with Leucas aspera might have something. We never knew that ultimately Leucas aspera is good like anisophyllin. We never knew that. That portion has been done by the Dr. Jaishankar. I have not done that portion, but another way, it is not a toxic plant. Plant is edible plant. People are taking it, and they found that it is a good plant for their health. So toxicity is not there.
Sir, adding to my question, sir, whenever we take natural active compounds, sir, sometimes what we analyze that if we are studying in vitro and in vivo, sometimes their characteristic change. Like if we are studying same compound and taking in vitro study, the characteristic changes in vivo. So, sir, do you have any idea how to deal with this mishap?
No, mishap occurs because of our something wrong that goes with our work. But whether it is a toxicity or not, that has to be tested first if you want to have a drug. That is first word. That you have to test whether it is toxic or not. Otherwise, you should not proceed. Coming with a toxic material is no good.
Okay, sir,
so
thank you.
That effect should be done. First of all, you do it with the mice or rat, whatever it is. You test them that whether toxicity is there or not. Toxicity test is primary. Must have been taken first.
Hello sir, thank you for such a nice talk. I have a genuine question. As you rightly pointed in the beginning that it's now a pandemic. Earlier we used to see our grandparents and parents being affected with this disease. Now even cousins or friends of friends. And for instance, even infants are being just diagnosed with the disease. So do you see this condition in those who are having it in early 20s or in infants, and the intervention that you make with anisophyllin A, will that work there as well for them as well? Thank you.
Thank you for this question, but we believe that anisophyllin A will be highly attracted. So we have to patent it. It's an Indian thing that Dr. Jaishankar has come out. And we tested it now testing everywhere, that is true. But one thing is this anisophyllin A, if it is taken by other companies the companies are always looking for this kind of drug. But we have to have the patent. We have now applying for the patent. We have make a secret out of it. A structure I have already shown to you. It's a huge structure. So this structure, nobody can do the anisophyllin synthetic preparation. That is not possible. Jaishankar was telling me that. Another thing is that Anisophyllin has not come to the market, not come to the anywhere, not come to the doctors anymore. This is the thing, the story we have told you. We are now trying to come up as a drug. That will take a lot of time. But we'll definitely come for a drug. We'll definitely come for a drug. Till now, it is done with the mice, with this model, but we have to come for coming to the bigger animal and see what happens. And for that purpose, we are also looking for pig, whether it is working or not. We are doing all these things, 4 or 5 people. Let us see what happens. We believe by next year we will be able to come. It's not easy to come as a drug. So.
Thank you, sir, for such a valuable and informative lecture. I now invite Dr. M.K. Arunasri to kindly deliver the vote of thanks.
Good evening, everyone. So I'm here to propose vote of thanks. I first of all thank our Vice Chancellor, Professor B.J. Rao, for his support to extend this distinguished lecture by Professor Samir Bhattacharya today. Then I also thank IOE Directorate for funding this distinguished lecture. Then I thank the office staff from PRO's office, VC's office, and Dean's office for their support, without whom I think it's not possible to organize such a lecture. And finally, I thank all the faculty, students of SLS and other university fraternity for gracing this occasion. Thank you all.